I want you to appreciate just the big picture first. The big picture is we're going to have amino acid pool coming either from the dietary amino acids, from cellular protein degradation, or synthesis of non-essential amino acids. That amino acid pool mainly goes here for cellular proteins, about 80%. Second component goes into specialized products. That includes neurotransmitters, nucleotides, heme, and so on, other important nitrogen-containing molecules. And the third component is you break down the amino acid to its carbon skeletons. And that's what we did there before. You do transaminases, urea cycle, you produce carbon skeletons on the side of from here. And these carbon skeletons can then be used as a source of energy, either to make glucose or to make other molecules like ketone bodies. All aminotransferases work on an amino acid. They'll remove the amine group, transfer it to an acceptor keto acid. And as I mentioned to you, one of the important acceptor keto acids is alpha ketoglutarate. So when you add the amine group to alpha ketoglutarate, you're going to form glutamate, the amino acid glutamate. So in many ways, many amino acids will eventually donate that nitrogen to alpha ketoglutarate to give you glutamate. So glutamate becomes the sink for all that nitrogen coming from other amino acids. Okay? The purpose of the urea cycle is to remove nitrogen / ammonia from cells, from the body, in a non-toxic form, say detoxifying ammonia, removing those nitrogen in a non-toxic form. So that's what urea does, the urea cycle. It'll incorporate two nitrogens in a molecule called urea. Urea is non-toxic. You may think otherwise, but urea is non-toxic. It can be transported in the bloodstream and excreted by the kidneys. Okay, that's the first important thing to think about. The purpose of the urea cycle, therefore, is to dispose of nitrogen in a non-toxic way. Here are all the enzymes that you want to know. The first enzyme is by far the most important enzyme, and it's the rate-limiting step, the rate-limiting enzyme. Carbonyl phosphate synthase 1 requires CO2, ammonia, and 2 ATPs to form carbonyl phosphate. Second enzyme is ornithine transcarbamylase. That condenses ornithine with carbonyl phosphate to form citrulline. Citrulline is now transported to the cytosol. In the cytosol, it condenses with aspartate. Aspartate is incorporated into citrulline to give you arginosuccinate. The enzyme is arginosuccinate synthase. Then arginosuccinate synthase. So aspartate is the second source of nitrogen. Those are the only two things that can get into the urea cycle. Either ammonia or nitrogen in the form of aspartate. No other forms of nitrogen can enter the urea cycle. Okay? Arginosuccinate is broken down to arginine by arginosuccinate lyase, releasing the carbon skeleton from here. If fumarate. This is just to tell you that really the fumarate is recycled. Don't worry about all the details here, but it's recycled. It's not wasted. It's used to make malate or oxaloacetate and so on. Don't worry about the details here, but just it's recycled. Arginine formed by arginosuccinate lyase is broken down by arginase, which produces urea and ornithine, and ornithine is now transported back to the mitochondria, and the cycle can begin again with another molecule of carbonyl phosphate. Okay? So know what enzymes are in the mitochondria, what enzymes are in the cytoplasm, and the sequence. Okay, the important allosteric regulator of the first enzyme is called N-acetylglutamate, and it's simply an acetylation, adding an acetyl group to glutamate. It's made by an enzyme called N-acetylglutamate synthase. This is a very important enzyme because it is going to provide this allosteric activator of CPS1, okay? Carbonyl phosphate synthase 1, which is the rate-limiting enzyme of the urea cycle. So N-acetylglutamate, here's the synthase again. It's actually activated by two things. The formation of this is activated by arginine and or glutamate. Glutamate is the substrate. Arginine can activate this enzyme that makes an acetylglutamate. So both of these are necessary, or really both of them will activate the formation of an acetylglutamate. And you can think about that after a protein-rich meal, you will have plenty of amino acids in that protein-rich meal, including arginine and glutamate. So you will form an acetylglutamate, and then that will activate the rate-limiting enzyme CPS1. Okay, that's the key step there, the first important regulator. And as I mentioned, if you have a protein-rich meal, you're going to have enough arginine and enough glutamate. The third important thing is this point here. The third point there is that all the enzymes of the urea cycle can be induced or upregulated. So if someone is, say, on a high protein diet, their urea cycle enzymes in the liver are upregulated. There will be more of those enzymes in the liver. Okay? If you like this video, please subscribe. Thank you.